All right, wonderful. All right, let's get started. So welcome to the afternoon session. So this session, we're going to have some deep dive discussions into how to handle data, how to tokenize it, and not only deal with how to do it, but also talk about practical issues when we try to scale up foundation models. So this is going to be done jointly by the Argonne and the team. And yeah, every speaker, please, instead of just jumping into this tutorial, please spend a couple of minutes talking about your background and what you've been doing. So I think that'll be useful as well, right? So Alex, start.
Sure. Thanks, Karthik, for the introduction. So my name is Alex Brace. I'm a PhD student at University of Chicago under Ian Foster and Arvind Ramanathan. And I do a lot of work at Argonne. I've been working at Argonne probably since 2020. We started working on COVID. And one thing you may or may not be familiar with is the GenSlim model. So this was the first large language model for a foundation model approach for scaling out to full genomes. And so that's a project we've been working quite hard at improving over the last year or two. And in addition to that, I've worked a lot on HPC applications, so starting up dynamic workflow ensembles that help to manage simulation campaigns. So, molecular dynamics, we're trying to understand biomolecular structures and functions and how they change in the cellular environment. And so we run massive simulations across HPC resources. And so we use AI to steer those to better sample the rare events. So I have a lot of experience in that, applying AI to science problems and in foundation models. So I think maybe I think some of my background will also show in the introduction to the Omics tutorial. So maybe without further ado, we jump into that. Can everyone see the notebook if I have it like this? Is the size okay?
Yes, you can maybe increase the size a little bit, but generally it's okay.
Great. And the Collab's interactive and it should all run for everyone. But if the screen is too dark on the screen share for whatever reason, I don't know if it's being broadcast, we can also look at the white background on the GitHub. So if there's a preference, otherwise I'll use the Collab.
Screen looks okay. Yeah.
Okay, great. So yeah, if you open it up, just let this run. This shouldn't take long, but we'll let that run in the background while we get into some background here. So like I said, we've been interested in genome scale foundation models. So what I'd like to do in this tutorial is maybe give some background on the biology and bring folks up to speed on what does it take, what sort of domain expertise does it take to actually build a foundation model in a specific scientific domain. First, we're focusing on genomics, but some of these lessons may generalize further. So we're going to go through, what does it mean to do redundancy removal of a training corpus for non-textual data. So this sort of omics is this branch of or branches of science that have been sort of being developed for a while, and it is mainly sort of interested in biology and how big data can impact biology. So there's many sort of the omics suffix is used quite broadly. We have genomics, proteomics, metabolomics, metagenomics, transcriptomics, all these omics. And what they essentially mean is they're modalities of data that have some scientific importance within biology, and they're large collections of this data. So you can see, this is sort of a basic diagram of, you might have your genome encoded in the chromosomes, and this genome, is essentially encoded in DNA. And this DNA, so we have massive amounts of DNA that we can sequence from genomes. This DNA codes for proteins. We can look at, what percentage of, the cell is composed of certain proteins. So considering all the proteins that are active in the cell, what is the community of cells? How do these communities form sort of tissues and larger, biological structures? And all this is kind of covered within this realm of omics. So it sort of refers to this totality of sorts. And so there are many types of omics, like I mentioned. I was on the Wikipedia page. There's actually 194 types of omics that they list. Everything from epigenomics to the exposome. So what environmental aspects are we exposed to that may affect our health? So this is all covered. So maybe why should you be aware of omics, or in other words, why do we think it's important? So we think this is sort of a central approach or perspective that we can take for understanding biological systems, and then applying that in a technical sense to improve life. So one aspect is personalized medicine, which you may have heard of, which is this emerging practice that perhaps medicine can be tailored to individuals based on their genetic profiles or based on specific features of what makes them unique. And so perhaps the sort of if you think of the average medicine, maybe it's not sort of tailored to an individual. So perhaps using omics, we can come up with useful descriptors or features that better inform the development of medicines for individuals. This sort of builds into this One Health perspective, which is sort of this overarching integrated approach that says, the health of humans and animals is also a function of the environment. So we really need all these aspects of our world to be sort of healthy in order to prevent, predict, detect global health threats such as, COVID-19 or other, viruses with pandemic potential or other environmental pathogens. So, omics helps look at this, big data and understand from this larger perspective what might be happening within the world. And similarly, we can apply this to sustainability. So we can look at, drivers of biodiversity loss, invasive species, pollution, how's urbanization affecting wildlife and humans, and as well as climate change. So omics helps provide data types and sort of ways of understanding data for all these areas. So it's quite fundamental. Now, like I said earlier, we've been building genome scale language models. So why do we apply machine learning, and what's the buzz around AI? Well, you can see this nice chart. So this shows the number of sequences that have actually been sequenced from the wild, and you can see that over time, in fact, since 2015, we've had this exponential growth of sequences in our databases, and this is e to the 9, so I think that's a billion. So about 5 billion sequences according to this plot. And so this is, of course, way beyond the scale that any humans or any specialists can process. So we need to rely on some sort of computational techniques. We've been developing machine learning techniques to, explore this data, develop new hypotheses, perhaps make new discoveries, and also on the engineering side, which is, we'd like to improve the quality of life on Earth, given the information that's stored here. And this is just for genomics, right, which is the focus of this tutorial, but you can see this similar sort of trend happening across omics. So there's a huge potential for data. So that's some background on omics. What I'd like to do is just provide some background on basic biology. So this is sort of maybe the computer science perspective of what do you need to know if you want to go about training a foundation model. So perhaps it's not so simple as downloading the data and spinning up the training code on a big compute cluster. There's some domain expertise required. So if you'll indulge me, we'll go over some definitions. So the first one, of course, is DNA. And this is basically the molecule that carries the genetic instructions of life. And so it's sort of deceptively simple. It's written in these sort of 3 bases. That's A, T, C, and G, adenine, thymine, cytosine, and guanine. And it basically forms this double helix structure. And this is, what we call like the language of life. It's encoded across, all prokaryotes and eukaryotes, and even in many viruses. They all have this DNA that encodes many things, and of particular importance, it encodes a set of functional proteins that eventually go out and do the cell's business that maintains the organism. So the RNA is another form of information storage molecule or information representation that is essentially it's a copy of the DNA such that copy can then be sort of distributed throughout the cell for the production of proteins. So the DNA, we sort of keep that in the nucleus in eukaryotes. We keep it sort of safe stored in there. We make copies of it. Those copies then go out, into the wider region of the cell, and there's these mechanisms that are called ribosomes that are responsible for converting that into proteins. And so maybe you train your foundation model on DNA, maybe you train it on RNA. There's not a great, there's not much to distinguish between those two data modalities in current foundation models. So that's definitely an interesting area for future work. And there's many types of RNA. There's this messenger RNA, which is that information that carries the protein instructions for making a protein. There's tRNA, which is a bit more functional. Those help assign amino acids to actually build the protein chain. We'll look at that in a minute. And then there's this ribosomal RNA. So RNA can not only form, not only be an information storage molecule, but a functional molecule. So it's quite interesting. And ultimately, this protein business is what the RNA is, or mRNA is building or encoding. And proteins, like I said, they're the sort of workhorse, or the little engines that do most of the molecular processes within our cells and throughout life. And so if you've heard of AlphaFold, this is a large AI model that is essentially solving this problem of how do we convert a string of amino acids. So there's 20 sort of canonical amino acids. And how do we take the string of amino acids that's encoded by the mRNA and fold it into this protein structure that has all this sort of complex topology and chemical bonding between different regions of the amino acids. And then furthermore, how do we consider what's called quaternary structure, which is combined multimers of smaller proteins that are responsible for larger functions within the cell. And so there's several levels of organization here. And what I want you to start thinking is towards this sort of hierarchy of how the information is encoded. It starts in the DNA, goes through RNA, goes through protein, and the protein has many levels of structure as well. And all these are sort of good candidates for modeling with AI. And of course, I sort of alluded to the ribosome as the molecule responsible for, or the structure responsible for putting this together into a valid protein chain. So it converts that input mRNA, matches up the amino acids that correspond. There's these little threemers, so groups of 3 bases, and those code for a particular amino acid, and that builds this sort of nascent chain that eventually folds into the 3D structure. Now this is getting a bit more bio 101. This is essentially, been broadly referred to as central dogma, or related to that, and it's essentially this recipe that DNA to RNA through ribosome gives me a protein. And this is what I would say the primary set of data modalities that we're interested with for genomic foundation models at this stage. And I'll just briefly touch, this is the genetic code. So if you've heard of this idea of genetic code, it's related to all of your DNA, but it's really this mapping of how does the DNA, the U's are the correspond to the T's in DNA. So that's the uracil gets swapped with the thymine in RNA. But these threemers, basically, so UCC would code for a serine molecule or serine amino acid. And this is the code that builds proteins. So that was the Bio 101 background. And now that we've seen some of that, we should actually be able to, start playing with this data. So just getting into basics of genomics file formats, we have this FASTA file, and I'll kind of breeze through some of this because it's pretty basic. You store some metadata descriptor, it's a plain text file, and it has this little caret to denote that header, and then you store the sequence, and you can store it on multiple lines. It's plain text, so very interpretable, and you can see that you could read the example FASTA file we have in this downloaded into this notebook. And you can actually print out the sequences that is contained within it. These are DNA sequences, and there's a function. So this is the BioPython library, and they provide some utilities to read these files as well as to translate the nucleotide into the amino acid. So you can see nucleotide sequence here and its corresponding amino acid sequence here. So I want to keep on track. But if anyone has any questions, I would say maybe we save them for the end, or do we want to take questions at any point?
Yeah, I think it's good to get questions as we go along because people might just lose their train of thought, right?
Yeah, so
Yeah.
any questions so far on the biology background or on the FASTA format?
No, I think the background was pretty succinct, at least for me. I mean, I've also been reading up a bit, so for me it was pretty straightforward.
Perfect. Okay, so let's talk about, and this is where things kind of get interesting, I would say. So let's talk about
So I do have a question on the data itself, right? So the sequencing that leads to all of this data, obviously that's not perfect, right? So there is errors, uncertainties. How significant are they, and is there an understanding of that?
Yeah, that's a good question. So yeah, the DNA sequencing technology, it's not my area of expertise, but I do recognize there's some complexities there. So you basically read the genome in multiple segments, and the idea is how do we compose these segments? And it's further compounded by the fact that a lot of times you're reading what's called a metagenomic sample, which is perhaps many organisms. So perhaps if you're reading them in this genomes in small segments, you could mix and match between different organisms when you're trying to concatenate the DNA. Certainly, that's a challenging problem, and I think one way we and we get into this a little bit. One way that's dealt with is by looking at sequence conservation. So, what are the samples I'm seeing similar to in our known databases, and that's how we can verify their what's in the sample and perhaps some of its quality. But it's certainly an open question, and it's one that probably machine learning can help with as well in terms of annotating.
Yeah. Any other questions from the audience? Please use the Q&A option on Zoom. Yeah. Keep going.
Great. So we want to talk about toward building a genomic foundation model. So I want to dwell on what do we want the foundation model to actually know, right? What do we want it to do? And for this, it's sort of important to understand the underlying data distribution and the process by which it's generated, which is evolution. So there's this notion of evolutionary conservation, which we just touched on in that question. And there's this important idea called sequence homology, which is essentially the similarity or homology between DNA, RNA, protein sequences. And this essentially defines, in most cases, a shared ancestry in this sort of evolutionary history of life. So two segments of DNA, there's sort of these three events that can happen that, this shared ancestry. So one's a speciation event, so I can branch off a new species, and perhaps the ancestor species I branch from, perhaps I share DNA with it or have a lot of DNA similarity. There's duplication events, which is a bit more complex, but the genomes can duplicate some information. And there's also this horizontal transfer between organisms that is especially prominent in bacterial systems that gives rise to, say, antimicrobial resistance and other traits that are conferred through plasmids and other mechanisms. But the point here is that there's some set of sequence motifs or subsequences that are shared throughout organisms, and those indicate, if we can track those, that basically tells us the evolutionary history of life. And so what we do here computationally is we define this notion of sequence similarity, and this provides us evidence of some common ancestral sequence, and we talk about alignments of multiple sequences. So we basically align the sequence strings such that we can see those common substrings And if you have a computational background, this sort of idea resembles the longest common subsequence problem. So essentially, if I give you a big string of DNA, what are the common elements of it? And the two most important takeaways here are that, sort of in bold here. So conservation indicates that a sequence or an important subsequence, which we call a motif, has been maintained by natural selection. It's evolutionarily conserved. And these patterns, right, along with the functions through the proteins that they encode, are what we really hope the genomic foundation model will learn. So we want to understand what are the common functions, what are the common motifs that build those functions, and if we can understand those, we might be able to do, some kind of engineering to design new proteins combining these motifs that perhaps do different things or catalyze reactions more efficiently, right? And you have a lot of applications that come out of that. So this is a very big discovery back in the 70s. And so just an example for why we care about evolutionary conservation. So I mentioned that RNA is actually sort of multifaceted in the roles it plays, codes for proteins, and it also has some sort of like structural element where it can do certain things in the cell. And so the ribosomes are actually made of this ribosomal RNA. And if you think about it, ribosomes are the mechanism responsible for converting DNA into proteins. So before we had proteins, we really needed to have ribosomes. And so those should be conserved across pretty much all domains of life. And so Carl Woese and George Fox actually did this analysis where they compared ribosomal RNA across different diverse group of species, and they actually saw that they could discover this new branch of life, or new domain of life called the archaea, by looking at these sort of methanobacterium organisms. And so you see a lot of the extremophiles, and not just extremophiles, but a lot of these sort of interesting organisms are actually in the separate branch of life, different from bacteria and different from eukaryotes like humans and plants. And so this just came from looking at those conserved regions of DNA. So this is the type of thing that we want these foundation models to learn an understanding of automatically. So any questions on that? I think there's a lot covered there.
Yeah, that's a lot of info. Would it be possible for you to drop the link to the Colab file or link to this, to the GitHub or wherever you have this, so people can look at this?
Yeah, so I'll post the link to this in the chat. So this is the main Git repo for this session and the tokenization section.
Yeah.
And the link to the Colab that we're looking at, I'll post here, actually.
Can you post in the general chat, or that's not possible because panelists are only a few?
Yeah. Thanks for that catch.
Yeah.
There you go.
If you quickly just go back to the co-op, I think for the people here, or go back to the GitHub.
Oh, yep.
That QR code, if you scan it, will take you right to the site. So that's an easier way for you to get there. Go down. You might need to zoom in a bit. It's GitHub SciFM intro to tokenizers. So it's a nice little, we got a good link there. But yeah.
All set? Need another minute?
Yeah, no, this is good.
Perfect. Okay, so that's evolutionary conservation, and that's basically the last big concept here, last biology concept. So now we'll get into a little bit more hands-on computational methods. And I guess, were there any questions before I move on the concepts?
Yeah, keep going.
Great. Yeah, so sequence alignment, right? I alluded to this longest common substring problem. Sequence alignment is a variant of that. It's a little more complex because we're not looking for just a subsequence, because perhaps we had some mutations that occurred, but it's still largely a similar sequence. So you can almost imagine like kind of a fuzzy match. And there's two ways that this has been thought about, well, two primary ways. One's a local alignment and one's called a global alignment. And they're exactly as they sound. So if I take a large sequence and I'm doing a local alignment, maybe I have a smaller sequence motif and I'd like to find where that maximally aligns within the sequence. Or just I'd like to find, if I have two longer sequences, I'd like to find their longest, sort of local alignment within them. The global alignment, on the other hand, sort of looks at, can I align the entirety of the sequences? So how can I optimally place sort of what we call gaps in order to optimally align the sequences? And we'll look at some examples of this to make this concrete. There's a few details here, but I just want to show some examples first. So I think this will become quite intuitive. So if we have these two sequences, right, you see ACCG, T, and then ACG. So intuitively, you might think, okay, there's an A that matches up here, and the CG tokens, maybe I zoom in a little bit, the CG tokens match right here. And so this is exactly what the alignment algorithm is trying to figure out. It's trying to score, these settings that I'm using here basically give a 1 point for an identical match and a 0 point otherwise. So it doesn't penalize these mismatches. And so you can see this aligned here. It put this gap in here. So perhaps there was an insertion mutation where a base was inserted into the sequence. And then you can see the CG is aligned here. And so the score is 3 because I have 3 matches. On the other hand, I could penalize adding those mismatches. So there's actually none in this example. So pardon me for that. But you can still see that if I set the match score to 2, I have 2 matches here. And the gap is not penalized at this point. So the score is 6. This 3 times 2 is 6. And you can see that this is another alignment that's equally likely, where I've aligned the AC and perhaps this C was a mutation instead of this one. So you can see the G is still aligned and this AC is aligned. So there's multiple potential sequence alignments, and we can look through those to see which is most likely. Now when I start penalizing gaps, so if I introduce a gap, maybe I penalize that by deducting 0.5 points each time I open a gap. So in this case, essentially have giving 2 points to the matches. So this is 6 points total, just like before. And I have 1 gap in here. So that's minus 0.5. And I have a gap here, which is a minus 0.5. So my score is 5. And so you can see playing with these parameters, and imagine doing this for much longer sequences, you can actually start to align the submotifs as well as the sort of these global sequence alignments. So the submotifs, very similar. You can see that this is a local alignment, and it's essentially aligning this region because it's the longest common subsequence within this particular sequence. And you can see this T was an insertion mutation, and this is not penalized because we're not including that gap penalty. On the other hand, I think, yeah, there's an example down here where if you include that gap penalty, you can see that there's 2 potential sequence alignments. So again, scoring 2 points for each match. So 1, 2, 3, 4, 5, 6 of those. And then I'm subtracting 1 point for a mismatch. And I'm also subtracting 0.5 points for the introduction of a gap. So you can see there's a gap here, there's a gap here, and otherwise there's these matches. And in this case, we have this dot here, which essentially indicates a mismatch. And this has the same score of subtracting this by 1. So that's the idea behind sequence alignment. And we can extend that to multiple sequence alignments. So this becomes more complicated. It's NP complete as far as aligning all these. But you can see this is more of a larger example. You have many of these protein sequences, and these colored regions are actually the conserved regions. And so these are the regions of the sequence that's conserved throughout evolution. And by comparing those, we can compare, which organism perhaps evolved from which other one, or what the common evolutionary relationships are. So any questions on the basics of sequence alignment? I skipped over a lot of details, but this is the basics.
So where you start the sequence matters, right? Or is it going to translate the two sequences such that there is some kind of match?
Yeah, so that's, you see that there's this gap token that's added here.
Yeah.
In this case. So yeah, it will move those sequence. It will insert those gaps, generally speaking, if there's sort of a better optimum within the alignment throughout the rest of the sequence.
Okay.
But you can see that there's some more amino acids for certain sequences, which is why we need those gaps. Yeah, and it's NP complete, right? So there's a bunch of heuristic algorithms for this. And intuitively, you can think of the large language model as learning, sort of, align these sequences. There's a sort of famous paper by Yashua Bengio that's jointly learning to align and translate, which is sort of the origin of the attention mechanism. And that alignment is key here, because it's learning what is the similarity of these sequences through distributional semantics when training over these massive corpuses of genomic data. Great. So I think here is where this is more of like a hands-on example. So just putting some pieces together, again, high level. And this is where it really relates back to building foundation models, which is this question of how do we do deduplication with respect to gene and protein data? You want to train foundation models across a diverse sets of data. However, there's a lot of bias within the sequence databases that sort of reflect what sequences humans care about, where's the funding, what organisms do we care about sequencing? For instance, we have a lot more SARS-CoV-2 genome sequence than for other viruses. So the question is, how can we ensure that the model training is not biased towards particular regions of the sequence space? And if we kind of think of this MSA, we can kind of say, okay, maybe we can cluster the sequences based on their similarity through alignment. And then when we're training, we can sample the training examples from each cluster as we go to sort of have a more diverse coverage over the clusters. And the way we're going to do that in this tutorial, or kind of think towards that, is using MMSeq2. I highly encourage you to check this out. It's a very cool method. We're not going to dive into its details, but it essentially, at a very high level, it uses alignments and some heuristics to cluster the similar sequences together. And it's sort of complex, so we don't have time to go into the details, but you can think of it as a heuristic algorithm for doing this clustering. And you need that for any massive scale approach. It absolutely needs heuristics. So we just, there's a bunch of code here, but what it is just providing these are just some custom utilities I made for reading and writing the sequences. So maybe an alternative to the BioPython example I showed earlier. And we have a function here, maybe provide a little more context, so just zoom out a little bit. This just runs the MMSeqs on Google Colab. So it's downloaded through Conda, and there's an executable. So we're just basically piping the information for the sequences we care about into this executable and running it as a sub-process, and then parsing those results into basically a pandas data frame. So the example we're going to go over is this protein sequence data from the dugong, which is a pretty cool marine animal. And we want to see how we can do some redundancy removal via clustering. And so if you're not aware of the dugong, they're sort of referred to as sea cows. They're last living, they're basically the last member of this family Dugongidae. and the other one went extinct in the 18th century. So yeah, they sort of roam around the seafloors, and they're quite interesting ecologically speaking. So what we have is this FASTA file that we've downloaded from Uniprot, which is this big database of protein sequence data. And we're just reading those sequences in. So there's about 160 of these. And then we're passing those to the MMSeq clustering. We kind of specify.
So that FASTA file has what, the entire genome of that animal?
It has
Of that
just
mammal?
the protein sequences that
Or
were
just all of the proteins associated with that?
Yeah, probably not even all the proteins, right? Because there is bias in the data set.
Okay.
So you wouldn't get all the proteins, but you get the coverage that have been researched and submitted to the database by humans.
Okay.
So that's inherently the bias in our data. And the question is, yeah, how do we, use the knowledge of evolution, of sequence conservation to sort of debias that for training the foundation models? So I won't get into these parameters, but these are the default parameters for MMSeqs, and they basically specify, the mode of sequence alignment, the sequence identity, or basically the % similarity between the sequences, and the essentially the coverage of the clusters. So the how are we going to, once we have the sequence similarity, how are we going to group those clusters together? What cutoff? And so you can see that if we run that, it's basically going to take the protein sequences and group them into sublists. So each list is going to contain a group of similar protein sequences. And we see that the number of clusters is 61 out of the 190 input sequences that were present in that FASTA file. So if we look, so you see that there's some redundancy in the data set already. And if we look at this histogram of the number of sequences per cluster, we see that the majority of sequences only have maybe less than 5 sequences within their clusters. So not too biased, but some of the proteins, there's, for instance, 12 very similar proteins within this data set. And so if we're training the foundation model without doing any consideration of this, we're going to get more representation of these protein sequences in the model, and this will bias the generation or the probability distribution towards this region. And we might also care about this region, so we want to debias. So the first thing we do is we get the largest cluster. So we just take the max over the lengths of these clusters. And we see that the size is indeed 12, so we've done it correctly. And these are basically like unique database IDs, session IDs of the protein sequences, just as a little printout. So we see what they are. And if we actually like loop over each of those sequences, and check that, so there's all the sequences in the FASTA file, and we check, hey, does that sequence ID, is that in the sort of that header tag? So we're printing out all the sequences in this cluster, and we see, okay, they're quite different. So we certainly wouldn't be able to see it by eye, but you also do see these common motifs, right? I'll draw your attention here, this LVII, LVII, and it continues, even though there's some difference throughout some of the other elements of the sequence. And if we actually look over to the metadata, we see that these are all hemoglobin subunits. So hemoglobin is a protein containing iron that facilitates the transfer of oxygen in the red blood cell. So it's kind of critical to mammalian life. And we can see that all of these protein sequences are, related to that function. And so these are sort of distributional duplicates in a sense, even though there is some uniqueness. If we look at similarly the second largest cluster, and print out the same thing, we see there's 7 sequences in it. And we see that this is all cytochrome B, which is actually, found in the membranes of aerobic cells. It helps mitochondria do its process for creating energy for the cell. And so you can see that there's, again, the sequence similarity. So this is how, if you're imagining training a model, maybe you sample, when you're forming your batch, you sample one of these sequences each time you hit this cluster, right? Instead of looking at all the sequences, and that'll help debias the model. So just as an exercise for offline, this Great Barrier Reef Foundation says that dugongs are more closely related to elephants than to whales and dolphins. And so in principle, you should be able to, you have the knowledge now to be able to prove this statement using some sort of sequence alignment and perhaps picking the right sequences to align to do that comparison. Perhaps cytochrome B is good, perhaps the hemoglobin, perhaps the ribosomal RNA would also work. So that's basically what I wanted to say. So we introduced omics data and some basic bioinformatics techniques that really gets at understanding the relationship between species. And this is what we really care about when we're building these foundation models, to get insight into, you know, evolutionary science, and then to sort of advance that further by designing, you know, new protein structures and perhaps new functions. So happy to take any questions. Thanks.
Thank you. So right now, what downstream tasks does Gen SLM do at this point?
Yeah, so the first iteration was really built for COVID in specific. And so we looked at, you know, basically, can we characterize variants of concern? So if we've trained it on the first year of the pandemic, how likely is the model to predict the next primary variant of concern? So that was our primary application. Now,
So more mathematically speaking, right? What does a variant of concern mean? Is it clustering?
Yeah, so variant of concern is essentially a set or you know, a set of sequence mutations that have arisen within the sort of population of SARS-CoV-2 that escape existing immunity in a way that is sort of worrying to the healthcare system and for human health. And so it's basically, you know, clusters of sequences that are related, but are distinct enough from the previous variants where, you know, existing vaccines or immunity may not cover them as well. So that's why we're interested in predicting those, or in just characterizing those from new data streams.
Okay.
So that was the primary task, and now we're moving towards more, you know, evolutionarily diverse. We're looking at training across large sets of bacterial coding sequences and beyond that. But those are still in development, so I won't say too much about it.
No, that's fine. No, just looking at it from not a domain perspective, right? From a mathematical task perspective. So if you did not have this foundation model, you would use a standard clustering technique.
Yep.
Right. So with this foundation model, what do you gain, or what does it bring to the table in that particular example?
Yeah, so for one, you know, of course it's generative. So this has a power all on its own. So we can actually not just look at existing sequences, but we can generate new sequences, or we can ask what's the probability of certain mutations, which is a better way to think about it than generating new sequences. So we can say, okay, here's the existing variant of concern, and what mutations are likely. And then through other, you know, bioinformatics and biophysical tools, we can say, what's the probability that those mutations that are sort of evolutionarily likely would escape existing immunity? So we can sort of have
that's very clear. So it's a likelihood computation, really.
It's a likelihood computation. You can also do clustering directly in the embedding space. So we refer to that as a semantic change score, but basically a cosine similarity or an L2 distance of the embeddings. And so you could imagine different strains are further away from others than they are within the strain, you know, with respect to sequences within the strain. So
okay, thanks. Any other questions from online or in the room? All right. All right. Thank you, Alex.
great. Yep. Thank you all.